day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Epigenetic Analysis Using Highly Validated Antibodies in Optimized ChIP Assays. My name is Andrew Jernan, and I'll be your X-Talks host for today. Now, today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using that chat panel. And at this time, all participants are in a listen-only mode, and please note that this event will be recorded and made available for future download. And at this point, I would like to thank Cell Signaling Technology, who helped develop the content for this presentation. Cell Signaling Technology is a private, family-owned company founded by scientists and dedicated to providing high-quality research tools to the biomedical research community. Their 400-plus employees operate worldwide, from headquarters in Danvers, Massachusetts, and offices in the Netherlands, China, and Japan. Now, I would like to introduce our speaker for today's event. Sayura Aoyagi received her Bachelor's of Science from Trinity College in Hartford, Connecticut, where as an undergraduate she examined bacterial single-stranded DNA binding protein DNA interactions using capillary electrophoresis. She then pursued her interest in understanding the complexities of protein DNA interactions at the University of Rochester in Rochester, New York, where she received her PhD in biochemistry in 2003. During this time, her work focused on the mechanisms of nucleosome remodeling from the ATP-dependent chromatin remodeling complexes SWITCH-SNF and MI2. This work was followed up by postdoctoral studies at the National Institutes of Environmental Health Sciences in Research Triangle Park, North Carolina. Here, she investigated chromatin regulation and the transcriptional mechanisms that govern progesterone and glucocorticoid receptor-mediated transcription. Sayura is currently an antibody validation scientist at Cell Signaling Technology, where she develops antibodies against targets related to epigenetics, chromatin regulation, RNA processing, and nuclear receptor signaling. And now, without further ado, I would like to hand the mic over to Sayura. Sayura, you can begin your presentation when ready.
Great. Thank you, Andrew. Thank you for that introduction, and thank you all for joining this webcast. So, as Andrew mentioned, I'd like to tell you a little bit about Cell Signaling Technology and the products that we offer. So what I'd like to do is to briefly remind you of what chromatin and epigenetics is and how in the recent years deregulation in epigenetics has been closely linked to diseases such as cancer. Then I'd like to talk to you about Cell Signaling Technology, CST, and how we are committed in providing the research community with great research tools, including antibodies, as well as optimized assays such as chromatin immunoprecipitation, or ChIP. And I'd like to make a note here that all of the products that I'll be talking about today is for research use only. So the chromatin is the way in which about 2 meters of DNA is stored in the cell nucleus. The basic subunit of chromatin is the nucleosome. And the nucleosome consists of 2 copies each of histone proteins H2A, H2B, H3, and H4, and about 200 base pairs of DNA wraps around that octamer histone proteins. These nucleosomes can then fold and compact into higher order architecture to further compact the DNA within the cell nucleus. Now, chromatin is not just a way to compact and store DNA. It plays an important role in regulating various DNA-directed activities such as DNA replication and repair, and of course transcription. So there are multiple ways in which cells can increase or decrease accessibility of important DNA elements to various transcription factors and RNA polymerase machinery. And I'd like to just talk about two mechanisms today. One is through the post-translational modification of histone proteins. So histone proteins have this N-terminal tail that sticks out of the nucleosome, which is relatively unstructured, and there are many sites of post-translational modifications, such as phosphorylation, methylation, and acetylation. Now, it's thought that these post-translational modification acts as a landing site for various chromatin-binding proteins that can either close up chromatin so that important DNA elements are now shielded from access to transcription factors and the polymerase machinery, thereby repressing transcription. Or these post-translational modifications can act as binding sites for chromatin factors that open up the chromatin architecture so that now important DNA elements are accessible to transcription factors and the polymerase machinery, leading to gene transcription. Another way in which accessibility of DNA can be modulated is through the activities of the ATP-dependent chromatin remodeling complexes, which utilizes the energy derived from ATP hydrolysis to alter histone DNA contacts, thereby increasing or decreasing important DNA binding sites to transacting factors. So these are just two of the many mechanisms that DNA accessibility can be modulated within chromatin. Now, it's become clear in the past number of years that deregulation of chromatin factors and epigenetic mechanisms are tightly linked to diseases such as cancer. So here's a list of some of the chromatin regulator subunits that are known to be recurrently altered in cancer, including subunits that are part of the histone post-translational modifiers, as well as the subunits of the ATP-dependent chromatin remodeling complexes that I described to you earlier. But recent years, researchers have also found mutations in histone genes themselves. So it's become clear that researchers really need to understand how deregulation of chromatin mechanisms leads to disease, what happens to gene transcription patterns when these chromatin regulators are altered, and how genomic stability might be altered, leading to disease. So in order to illustrate the point that epigenetic mechanisms and understanding disease has been key in the past few years, I wanted to focus your attention on a particular complex called the polycomb repressor complexes. So polycomb repressor complexes come in two parts, the PRC1 and PRC2 subunits. And it's involved in the transcriptional silencing of genes that's involved in differentiation, such as the Hox genes. Now, there are several different mechanisms by which polycomb repressor complexes have been known to or found to be transcriptionally silenced target genes. I just want to walk through one pathway, which occurs through the repression of transcription by trimethylation of histone lysine 27. Now, the trimethylation of H3 lysine 27 is catalyzed by EZH2, which is part of the PRC2 complex. And this trimethylation leads to subsequent recruitment of PRC1, leading to additional changes in chromatin structure and accessibility, leading to transcription repression. Now, EZH2, the subunit that catalyzes the trimethylation of this mark, has been found to be overexpressed and mutated in a wide range of cancers, including prostate, breast, uterine, and renal cell cancers. And EZH2 expression is often correlated with aggressiveness, metastasis, and poor prognosis. The way in which deregulation of polycomb is thought to play a role in disease is through the silencing of tumor suppressor genes and genes that promote differentiation and restrain proliferation. So there's been a lot of interest in understanding polycomb repressor complex function. But more interestingly, in the past few years, researchers have found that the target of this trimethylation, the lysine 27 residue of histone H3, can also be mutated. And this was found in about 60% of high-grade pediatric glioma cases. So in these patients, there is a lysine 27 to methionine mutation in one of the alleles of the variant histone H3.3. But when the authors decided to look at the total levels of di- and trimethylation of histone H3 lysine 27, either by Western blotting or immunofluorescence of cells that came from the patients with mutations, or comparing that against the wild-type neuronal stem cell, you can see a total global loss of these marks by these two methods, which was extremely interesting. Now, furthermore, the authors decided to look at the occupancy across the genome using ChIP sequencing, the lysine 827 trimethylation levels, and the enzyme that catalyzes the mark, EZH2. And what they've shown in this figure is, what they're showing is a sequencing track after ChIP sequencing. And what you can observe is a difference in association of the lysine 27 trimethyl mark in EZH2 when you compare the patient cell line, which is in red, versus the wild-type neuronal stem cell. And what you observe either at the chromosome level, chromosome 10 in figure A versus D, which is at individual gene level, you see that some locations that have high levels of H327 trimethyl in the wild type is diminished. For example, OLIG2 is an example of a gene that you can see that very clearly. But in some cases where there was very little H3K27, you're starting to see hotspots of the mark, which wasn't there in the wild type, which is also seen in the CDK6 gene. So what this suggests is that a mutation in H3 lysine 27 to methionine in one of the alleles of the variant histone H3.3 causes not only the global loss of total H3 lysine 27 trimethylation, but it changes the epigenetic landscape across the chromosome, implication being that this leads to changes in expression levels of important genes that are involved in various cancer pathways. And this leads to really important questions that one wants to answer. For example, how do those changes lead to disease? How is it linked to disease progression and initiation? And also you can ask really important questions such as how can you modulate these changes with drug therapy, for example, with EZH2 inhibitors. So incidentally, I just want to point out that this beautiful ChIP-seq experiments were done using antibodies that were made at CST for H3 lysine 27 trimethyl and EZH2. So I hope this example helped illustrate the importance of understanding epigenetic mechanisms and how it relates to disease. And in order for the field to move forward, we feel what researchers really need is high-quality reagents. So whether you're performing Western blots, IF, immunofluorescence, or IHC, you really need great antibodies that specifically seize the target, and it's very sensitive. And we feel that at CST, we can provide this. One of the missions of cell signaling technology is to be able to provide high-quality, highly validated antibodies to the research community across all applications. At CST, we extensively validate antibodies across all applications, not just Western blot, but IHC, IF, and ChIP, among others. And we look for specificity, sensitivity, and reproducibility of antibodies from lot to lot, which we know is really important for researchers to continue with their research long term. All of our products are produced and rigorously validated in-house by CST scientists such as myself for the consistent performance. So one of the ways in which CST is really committed to generating great antibodies is by developing rapid monoclonal antibodies. So in general, monoclonal antibodies tend to have better lot-to-lot consistency than polyclonals, but there are advantages to producing rapid monoclonal antibodies over mouse monoclonal antibodies, which is more common. One of the reasons is that rabbits elicit a stronger immune response, so it's really useful for generating antibodies against antigens with weak immunogenicity. Also, rabbit monoclonal antibodies don't have any IgG subclasses, and it tends to be a little bit more stable than mouse monoclonals. Also, rabbit monoclonals have higher binding affinities, affinities at the picomolar dissociation constant, and that allows us to generate antibodies that are very sensitive, but also very specific. And of course, rabbits are larger animals, and it produces 50 times more lymphocytes than the mouse spleen, which means that there's a larger number of monoclonal antibodies, candidate antibodies that we can choose from that seize different epitopes that work well for various applications. So it gives us more choice. But of course, generating rapid monoclonal antibodies isn't enough to generate great antibodies. These antibodies have to be validated to make sure that they specifically see the target that it's intended to see. So I'd like to give you several examples of how we go about validating our antibodies. So the first example I want to talk about is an antibody against BRM or BRAMA, which is a subunit of the ATP remodeling complex switch SNF. What you need to know about BRM is that it does have a homolog. There is a homolog called BRG1, and there is about 75% sequence identity between the two proteins, but they do have different biological functions. So when you're generating an antibody against this target, you want to make sure that it doesn't cross-react with BRG1. So we were able to test for this by using a cell line, NCCID, embryonic carcinoma, which is known to be negative in expression for BRM, but it expresses BRG1. As you can see on the left, on the Western blot, you don't see a signal, BRM signal in the NCCITs, as you would expect, and while you can see the BRG1 expression level in NCCIT, which tells us that our antibody is specific for BRM. We can also confirm this by immunofluorescence. So you can see in the HeLAs, you see a really nice nuclear localization away from the nucleoli as expected, and you see really no signal in the NCCITs. And of course, this is a chromatin associating protein, so one of the important applications for this antibody would be chromatin immunoprecipitation. So we've tested this on genes that respond to estrogen receptors or estrogen responsive genes, which are known targets of BRM. Now, sometimes we're fortunate that we have these known cell lines that we can use, or negative cell lines that we can use to validate our antibodies. But sometimes there isn't a known negative cell line, and we take this opportunity to use different model systems such as shRNA and knockout cells to validate our antibodies. So this example is validation of SIRT6 antibody, which is a member of the sirtuin histone deacetylase protein. So as you can see on the very left on the Western blot, the SIRT6 protein has really nice species cross-reactivity, sees both human, murine, and monkey SIRT6, which is great. But we wanted to confirm that our antibody is actually seeing SIRT6 by using first a shRNA knockout using HCT116. So compared to the non-targeted control on the left, in the SIRT6 knockdown, you can see a really nice diminishing of the signal when you use our antibody, confirming that our antibody is specific for SIRT6. Also, we've been able to validate SIRT6 reactivity in the murine cells in mouse using MAFs, either wild type or knockout, to make sure that the signal we're seeing in those lysates are real. And so as you can see, compared to the wild type and the knockout MAFs, you see complete loss of signal as you would expect. Again, validating this antibody for its specificity. And one last example I wanted to give in terms of validation here is validation against phosphorylation of CPhos at serine 32. In this case, what we did was to treat the cells with TPA, which induces phosphorylation at serine 32, as you can see from the Western blot. And this is completely lost when you treat the lysates with lambda phosphatase, which removes the phosphate group. We've been also able to confirm this by immunofluorescence. As you can see in HeLAs, you only see the signal with the addition of TPA in the nucleus, as you would expect, and this is completely lost in the presence of lambda phos. And again, CPhos is a transcription factor and an antibody that one would probably want to use for ChIP analysis, so we've also validated the antibody for ChIP, as you can see on the bottom right. So this talk is about epigenetics. I've given you some examples of how we validate antibodies, but we can't talk about chromatin regulation without talking about histone modifications. And as I mentioned in the introduction, histones can undergo various types of post-translational modifications as listed here. And generating an antibody that's specific against a certain modification is actually quite challenging. And we take a number of methods to make sure that when we're generating an antibody against histone modification that we're extensively testing it for its specificity. So I want to give you an example to illustrate this point. So one of the histone modifications that occurs on the histone tails is lysine methylation. So lysines can undergo different levels of methylation, either mono, di, or trimethylation. And biologically, these different levels of lysine methylation is not equivalent. So when you're making an antibody against lysine methylation, for example, trimethylation, you want to make sure that the antibody doesn't cross-react with the di or the mono version. What makes antibody validation a little bit more challenging is the surrounding residues around the lysine residue and their similarity. So, for example, if you're making an antibody against trimethylation of lysine 27, as seen here, if you look at the sequence surrounding that lysine, compare that against lysine 9, you can see the AR preceding the lysine followed by an S. So the sequence similarity around the lysine residue makes it really hard and challenging to generate an antibody that can specifically see lysine 27 trimethyl over the lysine 9 trimethyl. But when you are making an antibody against the lysine 27 trimethylation, you want to make sure that it doesn't cross-react because the trimethylation on lysine 9 and 27 are not biologically equivalent. And again, the antibody has to be specific. So in order to make sure that our antibodies are specific, we employ various types of assays, which are listed here. But for today's talk, I just wanted to focus on the peptide competition ELISA and the histone peptide array, with more focus on the histone peptide array. So the histone peptide arrays are simply glass slides with a nitrocellulose membrane on it. And on the membrane, we've spotted histone peptides with known post-translational modifications on them. And these are spotted at 3 different concentrations in duplicate. And we can apply the antibody that we're testing on these arrays to ask which peptides does our antibody see. So this assay is really important for us in terms of validating the specificity of our antibody. And I want to show you an example of how this works by talking about antibody validation against HV lysine 27 trimethylation. So as I mentioned, when we're making antibodies against lysine methylation, we want to make sure that it sees the specific level of methylation, in this case trimethylation, but not recognize di or mono or unmodified version of that site. And we can do that because the array has these peptides spotted on it. So we hope that our antibody only sees the trimethyl version of the peptides and not the others. Also, in the very same experiment, we can ask, does our antibody recognize lysine 9, which looks very similar to lysine 27 trimethylation? And we can answer that in the same experiment because the peptide for HV lysine 9 trimethylation is spotted on it, and hopefully our antibody doesn't see those spots. And then we can also use the array to characterize the antibody. For example, because N-terminal tail of histone are heavily post-translationally modified, neighboring residues could very well be modified as well. So for example, arginine 26 could be asymmetrically dimethylated. So we can ask, is the antibody still able to see the lysine trimethyl in the presence of arginine methyl 2A next door? And again, that particular peptide is on the array, so we can ask that question. And so once we apply the antibody on these peptide arrays, we can detect which peptide our antibodies are detecting by using the Lycor Odyssey imaging system, and this really allows us to quantitate the level of interaction of our antibodies with these various post-translational modifications. So here is an example of the data using our trimethylhistone HV lysine 27 rabbit monoclonal. The x-axis shows you all the peptides that's on the array. It's really not important that you can read all of it, but what I'd like to point out to you is that our antibody really only sees HV lysine 27 trimethyl, as you would expect, and there are peptides next to it that represent HV lysine 27 di, mono, and unmodified versions, and it really doesn't see any of that, which is great. This antibody is really specific. Also, what I like to point out is that the antibody can still recognize lysine 27 trimethylation in the presence of arginine 26 asymmetric dimethyl, which is an important information to know when you're interpreting the data as you obtain results from your experiments using this antibody. It's a type of information that I think is key in terms of data interpretation. So we feel that this peptide array is really powerful, and it provides a lot of information to the researchers using our antibodies. And so we'd like to make it available to researchers eventually. It's still not up on the website or the data sheet yet, but we'd like to change that and try to have that up as soon as possible. But what is in the data sheet is the peptide competition ELISA assay, which tells you the specificity of the antibody. So what you find in the data sheet is this type of result that I'm showing you on the slide, where in the peptide competition ELISA, the ELISA plate is coated with the lysine 27-trimethyl peptide, and the antibody is incubated at incubated with competing peptides at 2 different concentrations. And this could either be the target lysine 27-trimethyl, which will compete off the antibody away from the ELISA plate, thereby leading to decrease in the ELISA signal, as you can see from the green line. But if you incubate the antibody with any other peptide, for example, the unmodified version of lysine 27 or lysine 9-trimethyl, for example, it's not going to compete the antibody away from the ELISA plate, and you don't see a diminishing of signal, which tells you that this antibody is extremely specific for the intended target. So that's the data that you can find currently on our data sheets. Now, all of our antibodies, histone-modified antibodies, are at least lysine, acetyl, and methylated antibodies are validated by peptide arrays. But we don't just use the peptide arrays to validate products. We actively use these peptide arrays to develop our antibodies during the selection process in terms of being able to come up with an antibody that has the best specificity and also best utility for the researcher. And so I'd like to show you an example of that. So the example involves development of antibody against asymmetric dimethyl histone H3 at arginine 17. So arginine 17 on H3 can undergo asymmetric dimethylation catalyzed by CARM1, which is also called PRMT4. And this methylation event is helped by the prior acetylation of the next door residue, lysine 18, by P300 histone acetyltransferase. And it's thought that the activity of both P300 and CARM1 work together cooperatively to activate transcription. And there's data to suggest that in the cell, these 2 marks, the arginine methyl 2A and acetylation of lysine 18, occurs together. So when we're developing an antibody against the site, we want to make sure that the antibody can recognize it in the presence of lysine 18 acetylation. Of course, just like lysine methylation, there's different levels of methylation on arginines as well. It can undergo either monomethylation or dimethyl asymmetric, or I call it Me2A, versus dimethyl symmetric, or Me2S. And they're, again, not equivalent in terms of function, so we want to be able to generate an antibody that can distinguish between these different forms of methylation. So we use the peptide arrays to characterize some of our candidate antibodies to make sure that we're moving forward with an antibody that's specific. So here's an example of an antibody that has gone out of contention because, as the peptide array shows, it's not very specific for the arginine 17 Me2A mark. So you can see that it does see the arginine 17 Me2A mark, but it also sees a lot of the HC arginine 8, which is another site that can undergo asymmetric methylation. And it also sees HC arginine 8 methylation along with adjacent modifications next to it. So this type of antibody is something that we don't move forward with because it's not specific enough. But we have other candidates that we test, and this is an example of a top candidate that we would want to pursue for antibody development. So this antibody sees HC arginine 17 and Me2A, as you would expect, with very little cross-reactivity against any of the methyl sites known on the histones. And also, it recognizes the arginine 17 Me2A mark in the presence of lysine 18 acetylation. And if you remember, this is particularly important for this antibody because in the cell, these two marks are thought to occur together very often. And so this is really important in terms of thinking about the utility of the antibody. Now, this is in contrast to a competitor antibody that we tested. So in this case, this antibody is clearly very specific for the arginine 17 Me2A antibody, as you can see. It recognizes that peptide alone and really nothing else on the peptide array. But what you observe is that an obvious lack of signal coming from the arginine 17 Me2A in the presence of lysine 18 acetylation. And so this antibody can't recognize that mark in the presence of lysine 18 acetylation, which means that if you're using this antibody for various assays, such as ChIP sequencing, you're not necessarily going to get the wrong answer, but you might be missing a lot of data. And this type of information is really important for researchers when you're thinking about how to interpret the data correctly. So I've talked about various ways in which we validate our antibodies, both non-histone and histone antibodies. Now that you have access to these great antibodies, it's time to perform the experiments. And what CST does, in addition to providing these great antibodies, is provide optimized reagents and protocols for various types of assays that you might want to perform in the lab using our antibodies. And in today's talk, I'd like to focus your attention on ChIP, which is probably the most relevant assay to talk about when we're talking about chromatin regulation and epigenetics. So just so that we're all on the same page, I just wanted to briefly go over ChIP. This is a powerful research technique that's used to identify and analyze protein DNA interactions within the genome. And these factors can be things like transcription factors and co-activators, co-repressors, as well as histone modifications, among others. And the readout of ChIP is through single gene analysis, either PCR or quantitative PCR, qPCR, of DNA that the factors associate with. Also, more and more researchers are performing whole genome mapping, either chip-on-chip or more frequently now chip sequencing. So just briefly, chromatin immunoprecipitation involves fixation of protein DNA contacts, either histone or non-histone proteins, by formaldehyde. The cells are lysed, nuclei prepared, and the chromatin has to be fragmented. They're usually sheared or enzymatically digested to about 150 to 900 base pairs, and then the fragmented chromatin can be incubated with the antibody against the target you're looking for, whether it be histone proteins or non-histone proteins, and they are IP'd using protein gene agarose or magnetic beads. Once the immunoprecipitation takes place, crosslinks are reversed and the DNA can be purified, and the DNA is now ready for analysis either through PCR or through high throughput methods such as ChIP sequencing. So here's an example of the data that you can obtain by qPCR using antibody against trimethylhistone H3 lysine 4, which is a mark at active genes. So in these HeLa cells, you can see that this mark is prevalent on RPL30, which is a ribosomal protein, and GAPDH, as you would expect, because these genes are active in HeLa cells. And you see a really nice, robust signal above background, which is in red, which is the normal rapid IgG. And you see very little signal at MyoD1 and alpha satellite, which are silent genes in these cells. So I wanted to go over several factors that are necessary for successful ChIP experiments, and they are listed here. But I wanted to first focus your attention to the first two, which is having reliable, optimized protocol and high-quality reagents to do the assay, and being able to use these reagents to generate high-quality cross-linked and fragmented chromatin. So at CST, we feel that we're able to provide these 2 points through the SimpleChip kits that we have, the SimpleChip plus enzymatic chromatin IP kits. These kits contain all the buffers and reagents that's required for cell lysis, all the way down to DNA purification. And these kits come with complete detailed protocol from the very beginning, chromatin cross-linking all the way to PCR quantification, so that even if it's the first time you're performing the experiment, you'd be able to go through it quite easily. Unlike some of the commercially available antibodies from competitors, chromatin fragmentation is enzyme-based, which gives you great chromatin integrity, which we feel is important for a ChIP kit. And then DNA purification columns are included, so you don't have to buy outside vendor DNA purification columns or perform sort of dirty phenylchloroform extraction assays in the lab. And the kit contains universal positive control Histone H3 rabbit monoclonal antibody that you can use to make sure that you're following the protocols correctly, and it allows you to make sure that, especially if you're having sort of problems with your assay, you can troubleshoot your assay using the Histone H3 rabbit monoclonal antibody because it's everywhere in the genome. And it also contains a negative control normal rabbit IgG, which acts as the background control for your assays. And it also contains positive control RPL30 primer set. So again, coupled with the universal control antibody, you can really troubleshoot your experiments if you're having issues following the protocol. The kit is really versatile in that it works with both tissue culture cells and also whole tissue samples. So we feel that we have a really great product here that really allows you to perform ChIP effectively and get great results. But we also, to confirm that, we wanted to compare our SimpleChip kit results to those of the competitors. So in this competitor kit comparison assay, what we did was to look at several factors, chromatin-associated factors, and used our SimpleChip kit versus a popular ChIP kit that's out on the market and ask which one works better. One of the main differences between the competitor kit versus ours is that our kit uses digestion by micrococcal nuclease of chromatin at low detergent buffer, which preserves epitopes quite well, we think. And this is in comparison to the competitor antibody that uses sonication, where the chromatin is sheared by multiple rounds of sonication in high SDS buffer, usually 1% SDS buffer. So we wanted to compare our kit versus the popular kit that's used in the field. As you can see from the agarose gel at the bottom of the slide, for both kits you see fragmentation of chromatin ranging from about 150 base pairs to about 700 base pairs. And so this chromatin was used to perform the chromatin immunoprecipitation assay that I will show you next. So in this assay, we've used our kit versus the competitor using antibody against the RPB1, C-terminal domain of RPB1, which is the RNA polymerase, and also antibodies against two histone modifications, lysine 4 trimethylation and lysine 27 trimethylation. Just to orient you to this graph, the solid bar represents data, chromatin IP data obtained from our SimpleChip kit versus the lighter bar, which is the equivalent loci qPCR data that was obtained after chipping with the competitor kit. And as you can see from this data, for every antibody that we've used, we're seeing about 2 to 3-fold better enrichment of signal compared to the competitor when you use our SimpleChip kits. And this is really great data. You can still get great, you get great ChIP signal using our kit when you're using antibodies against RNA polymerase or histone modifications, which make extensive protein-DNA interactions. But what about when you start trying to perform ChIP experiments against targets that don't make as extensive of a contact with DNA, such as co-activators and co-repressors? So we wanted to test our SimpleChip kit against the competitor using antibodies against that type of targets. Here I'm showing you data for EZH2 and SUS12, which are the factors that are part of the polycomb complex that I talked to you about earlier. Here you now start to see a much more dramatic difference between the data you obtained with our kit versus the competitors. So the difference in signal to noise ratio is much greater when you compare the solid bar versus the lighter bar at known loci Hox genes for both of these targets. So we feel that our SimpleChip enzymatic kit really makes a difference when you start talking about performing ChIP against proteins that don't make as tight of a contact with DNA. So at this point, we have a poll question for you. We're interested in some questions about ChIPs, so please go ahead.
That's right. Thank you so much for that, Sayura. We certainly do have a poll question appearing on audience members' screens right now, and you can vote on any of the answers there appearing on your screen by clicking on them. The question we have for you, what type of protein-DNA interaction are you interested in examining with ChIP? Your options available are histone, non-histone, so for example transcription factors, both of those options, or neither of those options. That question again, what type of protein-DNA interaction are you interested in examining with ChIP? All right, looks like most of your votes are in right now. You can get your last clicks in because I'm going to be closing polling and sharing the results with everyone. Let's take a look. All right, there we have it. 48% of you said both. 43% said non-histone, so for example transcription factors. 9% of you said histone, and none of you said neither. And with that, Sayura, I'll hand the mic back to you.
Great, thank you. So it looks like a lot of people are interested in looking at both histone and non-histone targets. So I think our SimpleChip kits are going to work great for whatever ChIP targets you're looking for, and especially for non-histone targets that a lot of you are interested in. So I'd definitely give them a go. Okay, so now that you have great optimized protocol and high quality reagents to perform the ChIP assays, now you're actually ready to do the assays. Again, one of the things that you really need to perform a great ChIP is to have high quality ChIP validated antibody. So I've already spent quite a bit of time talking about validating antibodies for specificity, but just because an antibody works great for Western blotting or any other application, it doesn't mean that it works for ChIP. And so you have to make sure that the antibody you're using works in ChIP specifically. And we do validate our ChIP antibodies in-house to make sure that they work great in this assay. And we do this using proper controls as you would normally do in a lab with robust detection system. So here is an example. This is an antibody validation for ChIP using the glucocorticoid receptor antibody that we have. For this validation, we used A549 lung carcinoma cells and asked whether glucocorticoid receptors can bind to target genes in the presence of dexamethasone, which is the hormone that GR responds to. As you can see, you can really see a nice robust signal at SLC19A2 and MT2A genes, which are known target genes, in the presence of hormone, but not in the absence of hormone. And the signal is very robust compared to the normal IgG control, which is the red bar, which is the background control that we perform for all our validations. And also, the signal is specific for these two genes and not on the alpha satellite, which is the negative loci. So even in the presence of hormone, you don't expect GR to bind at that site. And so this assay really validates the fact that the ChIP signal you're seeing is from the glucocorticoid receptor. Another example of ChIP validation is an example here shown for antibody against NRF2, which is a transcription factor. And as you can see in the wild-type maps, you can see robust signaling ChIP signal at MAFG and NQ01 over the background control in red. And you see very low signal at RPL30, which is a negative loci, as you would expect. And when you perform the same ChIP assay using the knockout maps, since the protein is no longer expressed in these cells, you see no ChIP signal at any of the loci tested. And this is another way in which we can validate our ChIP antibodies. But it's not enough to just make sure that our antibodies can perform pull down DNA and is specific. We also go one step further to enable researchers to be able to pick up an antibody from our shelf and be able to perform ChIP experiments right away. And one of the important things that we do is optimizing our antibody concentrations for ChIP. So in a ChIP assay, it's adding more and more antibody doesn't necessarily mean that you'll get better signal. Usually there is an optimum dilution where you obtain the best signal to noise ratio. And so we try to optimize this for you experimentally ahead of time so you have a great starting point to perform your assay. So this is an example using an antibody against P53. And as you can see, titrating more and more of the antibody actually leads to decrease in signal over background. And in fact, the optimal dilution that we've determined for you is at 1 to 200. And so this is an information that's provided on the data sheet that's experimentally derived, so you have a really great starting point to use your antibodies at for your ChIP experiments. So I've spent quite a bit of time talking about ChIP, and most of the data, or all of the data that I've showed you, is ChIP qPCR. However, we do realize that more and more researchers are performing ChIP sequencing. It's a very powerful technique, and we want to make sure that our SimpleChip kits can perform and is compatible with ChIP sequencing analysis. So we have done this for the SimpleChip enzymatic chromatin IP kit as well as the Plus enzymatic chromatin IP kit using the protein G magnetic beads. You don't want to use the agarose version because it's blocked with sonicated salmon sperm DNA, so that might interfere with the sequencing. So you definitely want to use the magnetic versions. What we've done is to perform ChIP using our SimpleChip kits, purify the DNA, and then prepare the DNA library from the subsequent DNA isolation. You can do this with any prep kit that you like, but we use the NAB Next master kit or the Ultra DNA Library prep kit, and then performed next-gen sequencing. In our case, we used Illumina MiSeq or the NextSeq platforms. So here's an example of how our SimpleChip kit performs in a ChIP sequencing assay. We used antibodies against phospho-RPB1 antibodies, which are phospho-RNA polymerase antibodies, phosphorylation at different locations along the heptapeptide repeat, as well as the RNA polymerase N-terminal domain antibody, which is the control. As you can see, we see really robust signal across the gene, as you would expect, and the signal is really good, and the location and the specificity of signal across the gene is also as you would expect. So we feel very confident that our SimpleChip kit enzymatic chromatin IP kit works well for ChIP sequencing if that's the output that you're interested in obtaining. So here are a couple of concluding thoughts. What I've showed you hopefully today is that we have a lot of highly validated antibodies. They come pre-validated, optimized, and so it saves you time and money in terms of performing your experiments and having confidence in them. Also, we have a large portfolio of over 225 ChIP-validated antibodies, and these include about 170 of them are transcription factors that are not histone modifications, and it covers many research areas. Also, our company provides positive and negative control primers that you can purchase for every ChIP-validated antibody, so if you want to have a positive control for your ChIP assay, you can do that. We feel that our SimpleChip kits are superior to some of the competitors. It's low cost per assay, and the protocol is really easy to use, and it comes with the Histone HV Universal Positive Control Antibody, which helps you troubleshoot your experiment. And our kit yields more robust enrichment than some of the competitor kits that we've tested. And one of the things that makes EST special, I think, is that when you call in for technical support, you get to talk to someone who performs the assay daily so they can really understand some of the issues you might be facing in the lab, which I think really empowers your experiments. So with that, I'd like to conclude my talk. I just wanted to let you know that you can get a complete list of CSC ChIP products at the website link that's shown on the slide. The scientists who will answer your questions can give you advice about selecting antibodies and assist you in the usage of the ChIP kits. And the tech support for ChIP is shown here as well on this slide. And you're welcome to contact me as well if you have any questions about antibody validation. Thank you.
Fantastic. Well, thank you so much for that, Sayura. That was an excellent presentation. Lots of insightful information was presented there. Thank you. At this point, I would like to invite our audience to continue sending their questions and comments in right now for this, which is the Q&A portion of the webinar. And I've already received some questions, so let's get started. Our first question what is the minimum cell number required to use the SimpleChip kit?
Yeah, so I realize there's a lot of researchers interested in being able to use ChIP for low cell numbers. We've gone down as low as 400,000 cells for chromatin modifications. But for transcription factors, you really want to stay at around 4 million cells to get the optimum signal-to-noise ratio. So I would still stick with that number.
Okay, excellent. Thank you for that. Moving on to our next question here. This audience member says, I thought that monoclonal antibodies often do not work well in assays such as ChIP. Will I encounter these problems using your monoclonal antibodies?
Yeah, so we've heard this too, and in fact, we encounter that too. But I think a lot of people who sort of have encountered this was using mouse monoclonal antibodies, and in fact, we do sell some mouse monoclonal antibodies and validate them for ChIP, and we also find that a lot of the times the mouse monoclonals don't work very well for ChIP. There are exceptions, of course, like the P53 antibody that I showed in my talk, but this is not necessarily true for rabbit monoclonal antibodies. We find that rabbit monoclonal antibodies tend to work across all applications, not just ChIP. But also I want to point out that at CST, we start off with a number of candidates, actually a large number of candidate antibodies, and we specifically look for an antibody that works for the assay that's important. If an important assay is ChIP, then we make sure as we go through the candidates that we're picking the best one for that assay.
Excellent. Thank you for that. Moving on to another question here. How consistent were your qPCR results? This audience member says, I didn't see standard derivation or errors between the repeats. I think this was asked during your slide on antibody validation for ChIP with relevant model systems.
So you mean, I guess, experiment to experiment changes or differences in results. So the antibody actually gets tested throughout its development. So by the time you see the data, it's as a result of multiple experiments. And we make sure that it's repeatable. And also the antibodies undergo evaluation at a specific time level, like every year or something like that for ChIP. And so we make sure that the results are repeatable, and also we do that across different lots. So the results that we obtain are really consistent.
All right, excellent. Thank you for that. Moving on to another question here. Do all of your ChIP-validated antibodies work in ChIP-seq?
So we haven't, we're in the process of testing a lot of our antibodies for ChIP sequencing. We think that based on our in-house testing, as well as the publications that have used our antibodies for ChIP sequencing, that a lot of them are going to work. But since we haven't quite fully validated them and really come up with an optimal conditions for these antibodies, we can't guarantee that they work for ChIP sequencing, but I think a lot of them will work.
All right, excellent. Moving on to another question here. What about doing ChIP with mouse tissue? What is the recommended quantity for non-histone ChIP?
Right. So we have the SimpleChip Kit Plus kits have been tested using mouse tissue. In fact, if you go to our website, it gives you a list of different types of tissues and how much tissues you should probably start with in mgs. And also, I would still say ultimately you're using chromatin to IP, so you still want to get around 4 million cells or 10 micrograms of chromatin to do the IP. About 25 mgs of tissue per IP is usually good for most tissues, but it's tissue specific, and you can also still find that information on the website.
Fantastic, thanks for that. Moving on to our next question here. This audience member says, can I get access to the peptide array data before it's publicly available? And can I purchase the peptide arrays?
So the peptide arrays are not available for purchase currently. We don't have plans to sell them as of yet. But you definitely can get information, data on the ChIP peptide arrays. Just call the technical support, CST technical support, and ask for the data, and you will receive it.
All right, very good to know. Thank you again for that. Another question here, if a CST antibody is not recommended for ChIP, does it mean that it doesn't work at all?
No. So a lot of the times we don't recommend antibodies for ChIP because, for example, we've looked at a paper, we know that we think that a certain protein binds that location on the genome, but it doesn't necessarily work. But we don't necessarily have good positive controls that our treatment or cell model system is good. And so oftentimes the result is inconclusive. In those cases, we tend to be on the safer side and not recommend it. But that doesn't mean that the antibody is not going to work. It might have been the testing model issue. And so what I would say is that if you find an antibody that you like and you want to try it for ChIP, call the technical support, and they will be able to tell you if it actually has been tested in-house and if the test was a fail or inconclusive. When it's a fail, that means we had positive controls. We are very confident about the model system, so we can outright tell you that no, this antibody probably won't work for ChIP. But a lot of the times it's inconclusive, so we can tell you that. And if you think it's worth a try, you can definitely go for it.
All right, excellent. Well, thank you very much for those answers. We have reached the end of the question and answer portion of this webinar. If you do have any further questions, however, you can direct them to the email address showing on your screen for Sayura right now. That's sayura.aoyagi@cellsignal.com. Thank you, everyone, for participating in today's conference. You'll be receiving a follow-up email with access to the recorded archive for this event, and a survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our further webinars. Now, please join us in thanking our speaker for today, Dr. Sayura Aoyagi. We hope that you found this conference informative. Have a great day, everyone.